Good morning, friends. My name is Susanna Greer, and I'm the Chief Scientific Officer for the V Foundation. It gives me great pleasure to welcome you to the 2023 Answer for Cancer Symposia. We're going to start off this morning with a few words from one of our sponsors, ESPN. ESPN has been with us, as you know, from the beginning, always alongside us as a sponsor, but it's very special to ESPN to sponsor the Answer for Cancer Symposium. And joining us this morning is Kevin Martinez. Kevin leads the team that helps ESPN to accomplish serving the sports fan through purpose and commitment to cause, and builds the collaborative effort of sports changing the world. So please join me in welcoming Kevin.
Thank you very much. First of all, I have to say, if anybody was here last year, we promised Shane and I would do a dance for you. But we saw people dancing last night, and we're like, nah, we're not going to do that. So anyway, I hope you had a great time last night. This is a phenomenal event. Jane, Max, Shane, everybody putting this together. It is like literally the best in class and its ability. Thank you. Dr. Greer, thank you for your leadership. Thank you for your science. Science matters. And so I just wanted to share that with you, just a couple small things, and one give you a little bit of an announcement, which will hope make you very, very happy. First and foremost, you know, I have the privilege of leading a team that is at ESPN on the V Foundation, and a lot of people like, V, ESPN, why is that? Well, hopefully you all know Jimmy V's speech by now, right? But you also know that ESPN is built to use the power of sports for social good. So hopefully you see us and watch our Special Olympics broadcast, our work on veterans, the work that we believe is important with children's hospitals, also with the Walt Disney Company. We have celebrating our 150,000th wish. So as a corporation, and to be in my role, to have leadership that really, truly believes in doing the right thing the right way, it's a tough environment out there, but our leaders continue to say, do the right thing the right way. So we're so blessed to have the Walt Disney Company and ESPN lead us towards that energy. And I have an amazing team, Angela and Lisa. Please just say hello, raise your hands. They are phenomenal individuals. So, you know, I also just wanted to say, you know, our storytelling is what gets us there, right? So when we tell the Jim Valeno story, we tell the speech, we say don't give up, don't ever give up. We need to make sure it's right with sports fans, right? So we don't want to interrupt them in a live game. We want, you know, all this type of stuff. So we need to be really smart about how we tell the stories. And I hope you love our stories, because they are moments of greatness as it relates to journalism, but also as it relates to being able to get people to motivate to do something. So the call to action, I'm happy to announce today, that we have now helped raise over $200 million for the V Foundation with ESPN. So, and it's not just us, we don't take credit for it, but our sponsors, Rita Farrow is here from Disney Advertising Sales. Rita, we couldn't do it without the sponsors at the ESPYs, et cetera. So I just want to thank you. Jimmy Patero, who is a fearless leader in this space, who is also on the board of directors, who helps us every day. I can walk into any office at ESPN and say, I need something from the V, and I immediately get their attention. That you cannot get anywhere else in the world. Last thing I just wanted to share with you is in our storytelling, one of the great opportunities for us is to expand the audience, to get more people to tell the story. So I wanted to share with you, we have an amazing announcement. We launched it at the ESPYs, which is Hockey Fights Cancer from the NHL will now be powered by the V Foundation to raise millions of dollars for cancer research. So thank you. And we sponsor this because of the great science, because of the way the science is told. Thank you, Dr. Greer. Because remember, we got to talk to a lot of people like me out there that doesn't understand the science. So this really helps us get to the end result, which is what's happening with the dollars that are raised. So thank you so very much.
Thank you, Kevin, and thank you, ESPN. There's people in the back. Please come on up. We have chairs in the front. I see 4. I was a professor for a long time. No one ever liked to sit in the front row, so friends, come on up. We are in for a real treat today. I think if you are affiliated with the V Foundation, certainly if you work for the V Foundation, if you've ever heard of Jim Valvano, you have a favorite quote. My favorite quote is more of a story, and it's one that has probably become more family lore, and it's one that I heard first from my boss, Shane Jacobson, but then saw retold by Jim Valvano's brother, his little brother, Bob Valvano, on the ESPN 30 for 30 film Survive in Advance about Jim Valvano. So it was the story that was told about when Jim was little, and I don't know how young he was. I think he was around 17. I have a 17-year-old, so I can relate to this story, that Jim had big goals. And he wrote these goals down on 3x5 cards. And they were really kind of crazy things for a teenager to have, like, I want to have a Cadillac, and I want to be on the Tonight Show. And one of these goals was, I want to win a national championship. I mean, sports were a big theme in the Valvano family, and Jim's dad, Rocco, was a basketball coach at the time, and he knew what that meant to win a national championship. And so family legend has it that one day Jim's dad brought Jim upstairs, and he had a suitcase in his bedroom, and I can imagine if I did this to my son would be, what is this? And Rocco showed Jim this suitcase and explained what it meant. And he said, this suitcase means my bags are packed for you. I believe in you. If I have caught wind of the fact that you have written down goals, and that one of your goals is that you are going to win a national championship someday. You're going to be a basketball coach, and you're going to win a national championship. And he's saying this to a kid. He's like, if you believe it, I believe it. And later, as Bob Valvano and Jim Valvano told the stories of, how have you had these incredible successes? They said it was because our parents believed in us. And so how does that relate to why we are here today? So if you have ever given $1 to the V Foundation, you believe in us. And what the V Foundation believes in is research. We believe that research is going to change the path of cancer patients. And our bags are packed for the 3 individuals that are sitting on this front row. We believe in them. You are in for an incredible treat today. You're going to meet 3 of our grantees, and they are representing over 1,200 grantees that the V Foundation have invested in. We invest in high-risk research. It means it always doesn't work out, but when it works out, it's a home run. It's incredible. Our bags are packed. So today, I'm going to allow you a peek into the world that I see all the time, which is breathtaking. It's full of hope and joy, some frustration, but a lot of real excitement for cancer. So I can't wait for you to meet them. Today is going to roll out like this. We're going to have 4 presentations. The first is by someone who I cannot tell you how much I admire. She is the chair of our Scientific Advisory Committee. And in the audience today, we have all the other members of our Scientific Advisory Committee. If you don't mind, just raise your hands so they can see you. Their bags are packed as well. These are the thought leaders, the most prestigious cancer researchers in the world. And their bags are packed because they review the grants of all of the individuals who apply for cancer research funding from the V Foundation, and they manage somehow to sort out really impressive, really good grants and pull out those that are truly outstanding. So their bags are packed. So what's going to happen today is, first, we're going to hear from Dr. Davidson, who's going to help us understand what is cancer therapeutic resistance, lay the groundwork for us of where we've come, and why we still need you, why we need your bags to be packed for the V Foundation and for us to do more work. Where are the challenges? Why do we need to fund more research? Why do we need to help more grantees be launched by the V Foundation? And then we're going to hear from 3 of our grantees, and then we're going to follow that with a Q&A. And you can ask questions of any of our presenters and also from members of our Scientific Advisory Committee. So with no further ado, I'd like to introduce Dr. Nancy Davidson. Dr. Nancy Davidson is chair of the V Foundation Scientific Advisory Committee. She's also the executive vice president of the Fred Hutch Cancer Center. She's a professor of medicine at University of Washington, and she's the Raisback Chair in Collaborative Cancer Research. So please help me welcome Dr. Nancy Davidson.
Thank you so much. Kevin, this NHL thing is fantastic. I was at Pittsburgh as Cancer Center Director for a long time. Mario Lemieux, Hockey Fight Cancer, was wonderful. And I can't wait to see now, as a Kraken fan, how we're all going to be able to come together to support our mission. So thank you so much, ESPN and Kevin, for this newest relationship. Now, as you heard, I'm the more senior presenter today, and my job is to provide some perspective for the amazing work that you're going to hear from our colleagues who are funding as part of the V Foundation mission. So here's what I want to talk about. 30 years of progress against cancer, and we've made a lot, but also what are the remaining challenges, and that's, of course, why you're here. So this is not a new topic, right? I think those of you who've come here before have seen this slide from Bill Nelson. Thank you, Bill. I perpetuated it. Just remind us that this is not a new problem. Here is a report from the ancient Egyptian literature about a breast cancer, an unripe hemet fruit, a kind of fruit there which is hard and cool to the touch, no therapy. Now, I'm a breast cancer person. Of course, these days, I almost never see a breast cancer that comes in like this. Thank you. And there are a lot of therapies for breast cancer. So we've made so much progress on this ancient form of diseases. And let's look at that. I want to spend a moment thinking about some of those statistics. So here we're looking at cancer mortality, and we're going to look today in the U.S., and I realize that's a little U.S.-centric. We could have a whole discussion about cancer as an international problem, because it really is. But our time is short, so here are U.S. statistics. And just to remind you that we are making some progress. If you look along the horizontal axis, 1975 to 2020, and on the vertical axis, you see the rate of mortality per 100,000 of population, and you can see that we are slowly but steadily coming down ever since about 1990. Really major impact, both in men in the blue and women in the pink. Now, you can also think a little bit about some of the landmarks that go with that. Some of you in the audience probably remember when Richard Nixon signed the National Cancer Act in 1971, one of his major accomplishments as president, and something that I think really set in motion the focus that we have seen on cancer research and biomedical research actually over the last 50 plus years. So there we are in 1971 with President Nixon. Today, we're, of course, celebrating 30 years of the V Foundation, and you can see that blue arrow there, 1993, when Jimmy V gave his speech, and the foundation followed shortly thereafter. And I like to think maybe it's cause and effect, who knows? But you can see that those mortality slopes are beginning to lower. Right about 1990, 1995, we're beginning to see some of the impact of the work that's being done. Now, this has not been even. If you look at the top there, you can see in my little box the progress we've made at 3 different points in time, 1975 to 77, 95 to 97, 12 to 18, for all cancer sites in the US. And you can see a steady increase in 5-year survival rates from 49%, less than half, in the mid-70s, to now 2/3 of individuals as we come into the late 2012 to 2018. But it's also clear as you go down that we're doing better in some diseases than others. In this green boxes, you can see the cancers where we've had really quite a lot of impact, I would suggest. Breast cancer, my favorite, or my personal interest, 75% to 91% over that period of time for 5-year survival. Colorectal cancer, a lot of progress. Leukemias, we'll hear something about that today. Lung cancer, not fantastic, but from 12% to 23%, so that's a pretty major change, a doubling of survival over these years. Melanoma of the skin, and prostate cancer, of course, where we've also made tremendous progress. Sadly, though, this is not true for all cancers. Pancreas, Margaret Tempero's area of interest, we have made almost no progress, so much left to do there. And in some places, uterine cervix and uterine corpus, the uterine cancer, you can see that we might, if anything, be losing a little ground, and that's certainly horrifying to think that cervical cancer and uterine cancer, that the outcomes might be slightly worse now than they were a few years ago. So we have a lot left to do to try to think about some of these things, and that's, of course, why you're here. Now, over the years, we have developed, and you've heard about, if you're regulars here, a variety of ways of trying to tackle cancer. Surgery, one of our oldest modalities, dating back literally to ancient times almost. Radiotherapy, more than 100 years of experience now. The cytotoxic chemotherapies, which came out of World War II, and a lot of the developments that took place then. And then what has really captured us, and what is going to be a big focus today, are precision therapies, therapies that are based on our understanding of the biological capabilities of a cancer, and immunotherapy. So these are our newest modalities to bring to the table. Now, lots is known about how cancer is formed. That's actually been one of the big advances over the last 50 years, is understanding the process of cancer formation, carcinogenesis. Early on, it became clear that this is partly a genetic disorder, right? Peter Knoll was one of the individuals who showed us that acquired genetic defects, changes in the gene sequence, and the evolution of those changes over time led to the development of cancer and to its evolution and its progression. And you can see here in that little schematic, that normal cell, which in the course of its division will unfortunately incorporate genetic errors that may not be corrected. And with time, these can be propagated. They can lead to the cell growing in a more uncontrolled fashion, ultimately becoming metastatic, leaving its original source. And as we're going to talk about today, often becoming resistant to the treatments that we know how to employ. So this is the genetic evolution that we've been studying and thinking about for many years. Now, drug resistance is one form of resistance. There's also radiation resistance, so there are ways that cells can become resistant to many of the kinds of treatments that we have. But basically, just to remind you that we have that tumor on the left before treatment. And remember, it's a mishmash of cells. They're not all the same cell. They have different genetic capabilities. So some of them, those blue ones, are very sensitive to our initial therapy. But a handful of them, those red ones, might actually have characteristics that make them resistant. You apply your therapy, and so in the middle you can see that that tumor begins to shrink. But some of those red cells, those resistant cells, are now beginning to emerge. And with time, under the pressure of that therapy, those sensitive cells will perhaps go away, but some of those more resistant cells will now emerge. And so this is a substantial challenge for us when we think about trying to improve outcomes for cancer. Now, I have focused on DNA here for a moment, but let's just remember that cancer is not a disease only of DNA. It turns out that it has a variety of hallmarks. And I don't want to go around the entire circle, but to remind you that cancer cells live in a neighborhood, right? They have a lot of neighbors, a lot of different kind of cells that surround them, immune cells, blood cells, all sorts of cells that they need in order to maintain their health and their well-being. And so we've been able to unravel a lot of the pathways, a lot of those mechanisms that contribute to the carcinogenic process. And once you understand them, then you can think about how you're going to try to tackle them. So let's just pick a couple. Down at the bottom there, you know that cancers must develop vasculature, blood supply, if they're going to grow. Many cancers learn how to metastasize. They learn how to invade and go elsewhere in the body and set up housekeeping. They might be involved in inflammatory processes, which would be detrimental, and so on around that wheel, a variety of mechanisms that can contribute to the cancer growing process. and where if we understand them, we might be able to think about how we can impede them. And this work is not done. On the right, you see this wheel where we're beginning to think about even more mechanisms, more means that cancer cells might have at their disposal to develop into this resistant cancer and to grow. For example, there's a lot of interest right now in the relationship between our bodies and your microbiome, right? The vast number of organisms that live within you, and whether or not they might contribute in some fashion to the cancer process. We're interested in things like epigenetic phenomenon, so not just the genes, the DNA that changes, but other things that might change the DNA and how it interacts with other parts of the cell process. So a lot of opportunity here to try to think about what goes wrong, what can we do to overcome that, and what can we do to make sure that that cancer cell, if you shut down this mechanism, doesn't just switch over and move to a different mechanism. And that is the source of a lot of work. Very busy slide. My only point would be that we have thought very systematically about what are the potential mechanisms of therapeutic resistance for that cancer cell. You can see with that complex mix of cells living in its very complicated environment. So we can begin to think about how we tackle the growth of the tumor, how we tackle the burden, take out the large part of the tumor, for example, with surgery. How can we deal with the fact that this tumor is a mix of so many different kinds of cells, and so we have to think about how to address all of those cells? How can we think about the fact that, for example, for brain tumors, one of the problems is our therapies don't routinely, our drug therapies may not cross into the brain because of the blood-brain barrier. So lots of mechanisms that have been identified, and where, again, once you understand them, you can begin to think about how you might be able to circumvent them. I want to close by reminding you that it's not only about biology, that there are what I call social determinants of treatment resistance. And that is that no therapy is good if you don't get it, right? And unfortunately in our country and in other places around the world, there are a variety of disparities in cancer treatment. And here's just a list of them for each of the modalities. The one I would put down on several of them have to do with delays, that people who are in disadvantaged groups in our country may not have access to timely therapy in quite the way that other people would. Or in some cases, they may not even get it. At the bottom there, you can see for immunotherapy, an instance where Hispanic patients were less likely to receive immunotherapy than white patients for any of a variety of reasons. So I want to put that out, that there are not only biological sources of resistance that we have to think about, but there are what I would call social determinants of treatment resistance as well. Now, we're also now at another kind of inflection point. You know that President Biden has certainly made cancer one of the major features of his biomedical initiatives, and the President's cancer plan was reinvigorated earlier this year. It's a very ambitious goal, and I think we can all commit to it. The goal is to cut age-adjusted cancer death rates by 50% in this country over the next 25 years, and we would hope, of course, that those inroads would then spread around the world. That means we have to make what looks like a pretty small change. That slope, that 2.3% decrease in cancer mortality that we saw in those early graphs, we got to get it to 2.7%. That doesn't sound like very much, but actually that's quite a lot of work. There's a big lift there. And so that's where we all come in as we think about how we can help with these goals: preventing cancer, detecting cancers early, developing effective treatments, thinking about treatment resistance, our theme today, eliminating inequities, something that I've talked with you about, delivering optimal care, which is the same thing, engaging everyone, every single person's got to be involved in this, maximizing how we use all the data that is becoming available. And of course, we won't talk about it today, although this is one of our goals. We have to optimize our workforce. And you're going to see the future of our workforce today when you see the three speakers who follow me, who are going to be the people who are going to take this area and this research forward. So with that, as you say, Suzanne, everybody has their favorite Jimmy V quote. I like this one, which is, how do you go from where you are, and here's where we are today, 30th anniversary, to where you want to be? Where do we want to be 30 years from now? Well, of course, ideally, I'd like to be here drinking wine in Napa without talking about cancer, but We have to have an enthusiasm for this, and I hope that you will, a dream and a goal, and we all have to be willing to work for it, and I hope that you will continue to join us in this fight. Thank you very much for your attention.
Thank you so much, Dr. Davidson. All right, please join me on the stage, Dr. Katie Campbell. Dr. Campbell is a postdoctoral fellow at the University of California in Los Angeles. She's in the laboratory of Dr. Anthony Ribas at the Parker Institute, and she is supported by the Parker Institute for Cancer Immunotherapy, and she is a V Foundation Bridge Fellow. Please join me in welcoming Dr. Campbell.
It's an absolute honor to be here today. I would say for someone in my position, I'm what's known as a postdoctoral fellow. This is a very rare opportunity. It has really just reinforced how important the funding community is to advance a lot of the research, support the educational programming, and so I can just from the bottom of my heart for everybody here today, I'm just very grateful for this opportunity. So I'm going to talk a little bit about the project that I'm working on that is in part supported by the fellowships that I've received from the V Foundation, focusing on overcoming frontline therapeutic resistance in melanoma, and the way that we've accomplished this through translational research with clinical trials. So just to recap a little bit of Dr. Davidson's introduction, therapeutic resistance fundamentally means the tumor isn't responding. Our objective is to kill those tumor cells. And so for the audience here, I'm going to create the analogy that we're really trying to get past the defenders to make the shot, to make the basket. So if we're thinking about therapeutic resistance from a scientific perspective, we can say, why can't we score a basket? Or why is this happening? If the basket doesn't exist, if the drug target doesn't exist, you're never gonna make one because it's not there. If there's more defenders in front of the basket, sometimes there are just more tumor cells, sometimes that tumor is bigger, and so there's just more things we have to get through. This is sometimes a problem of dosing, sometimes it's a matter of competition, and so we have to account for this as we're trying to think about how the patient should be treated. Sometimes the defense is really good. They have a playbook as well, and sometimes they might be really tall. Here, that's also really important or helpful. But tumor cells, sometimes we say that they're smart, but they have, as was shown, they have really evolutionary ways to evade therapies. And so if those tumor cells find ways to avoid being detected by the drug or resist dying, we can't think about it in the same way. So fundamentally, in all of these experiments and all of these analyses, as we're thinking about the next step, how do we overcome this resistance? So we can focus on the defense, and a lot of our research is understanding these mechanisms, but we can also consider a better offense. And this is where immunotherapy has really changed the way that we treat, the way that we think about treating cancers, because we're not just trying to kill those tumor cells, we're actually harnessing the power of the immune system in order to build an offense that can see the cancer and can kill the cancer. So rather than that drug just getting inside of those cancer cells, hopefully killing all of them, we're trying to bring up a larger offense that can see and also eliminate those cells. So one class of immunotherapies includes anti-PD-1 therapies, and they have revolutionized melanoma treatment. With the approval of pembrolizumab back in 2014, and we have a large number of anti-PD-1 therapies, it's improved these response rates. So 41% of patients with melanoma are alive after 5 years. When I started my graduate training back in 2014, and people would talk about curing cancer, and honestly, I refrained from overemphasizing the word cure, but immunotherapy, the ability to eliminate those cancer cells, and the way that the immune system works is that it can remember, it has that memory and can continue to make sure that they don't come back. It really brought that word cure into that conversation. And this is an incredible, so this is a survival plot, and basically the higher the bar, the better the patients are doing. And so that y-axis is showing overall survival with time along the x-axis. And really we're looking at that 60-month mark where that 41% of patients are responding. But we really have to think about that 59% that aren't responding to. This is where we utilize clinical trials to say, how do we improve survival? How do we do better for patients? And so ipilimumab is another type of immunotherapy called an anti-CTLA-4 checkpoint blockade therapy that can help build a bigger T-cell offense. So it can really help the T cells get out of the lymph node and really introduce a larger offense. I know that there's rules about like numbers of players on the court and stuff, so bear with me. But really we're trying to get more of an offense so that we have less of a defense and more players probably statistically more likely to make a basket. But that being said, the SWOG Clinical Trial Consortium is a national clinical trial consortium as part of the NCI that with over 100 sites across the country, we enroll patients from community sites, and for this particular trial, SWOG 1616, patients with melanoma refractory to anti-PD-1, so patients that were treated with the standard of care therapy of anti-PD-1 therapy and did not respond, were enrolled into this trial, and with 94 patients across 34 sites across the country, and a large collaboration and consortium of clinicians, we were able to improve that progression-free survival by about 29%. So this paper is in press now. Yeah. So what those numbers mean is that from those 59% of patients that didn't respond to therapy up front, we have another 29% of patients that can continue delaying the progression of their disease. What that more time gives us more time, fundamentally, and it allows us to continue understanding how this is working and potentially how it's not. So I am not a clinician. My job was not to enroll patients. I don't really interface with patients very much. But my job is actually to focus on what's happening, how it's happening. And so this was a very, very large collaborative effort to collect blood and collect biopsies from these patients, and my job is to make sense of them. So this is a really busy slide. With an individual biopsy, we can do a lot of experiments from that. Those biopsies are essential to say the things that we study maybe in cell lines or in mice or in the lab setting. We have to look at these biopsies and say, is this consistent with what we're seeing clinically? And that's really what this translational medicine is doing. So from an individual biopsy, some of it goes into genomic sequencing. Many are more familiar with things like Ancestry and 23andMe. So tumors come from changes in the DNA or mutations. And so with genomic sequencing, I can look at it and instead of saying you're Scottish, Czech, and German, I think, and you can say this is the genetic ancestry of the tumor. This is what's driving its biology, and this is what may be wrong with those tumor cells. We also use pathologic review. We have a team of very talented dermatopathologists that look at each of these slides and look at the morphology of those cells on those biopsies. So oftentimes they look like large and pink and purple blobs. But really what the pathologists are saying is, does it look like these tumor cells have other things? What does that tumor microenvironment look like? And I'll show a couple examples of that in a second. And then finally, which is probably my favorite part, is the spatial profiling. We've come so far in technology that I talk about one experiment, but one of these experiments can give us tens or hundreds of thousands of features to look at these tumors. And so part of this whole process is to figure out what subset of those features allow us to understand how the drugs are working. And so with each of those biopsies, I think we collected close to, from the 84 patients, we collected about 300 biopsies from this entire clinical trial prior to therapy and while they're receiving therapy. And we were able to, within our lab, process all of these in order to do all of these analyses. So So these different types of analyses address different questions. So how are the tumor cells behaving? Because we know that these drugs are working by activating the immune system, we can say, how is the immune system responding? And are there immune cells seeming to get to the tumor cells? Because it's really important if the immune cells are there. It's like, in that way, the drug target might not be there, or the basket doesn't exist. We have to consider if that is working, and then also which patients that's working for. That allows us to more strategically move forward clinically to say these patients should get these therapies, and these are the patients that we need to identify other opportunities. So why is the defense so good? Is the offense scoring? Is the offense even advancing the ball? If the answer is no, obviously, we have to think about things a little bit differently. So it's been very exciting to work on this project, one, because the key is those on-therapy biopsies. What that is a snapshot of the tumor while things are happening. Either the drug is working or it's not, which we evaluate clinically through scans or, checking on the patients. But with those biopsies, I can also say, is it doing what we expected? Are these hypotheses being met? So here is an example of what's called immunohistochemistry. These slides are tumor biopsies really zoomed in, and each of those blue dots is a nucleus of a cell. The brown spots are things that are stained for T cells. So before therapy, this patient has next to no brown spots, whereas on therapy, after 1 month of therapy, we're able to see those T cells getting into the tumor. We're at a very exciting time from a technological space in that we can do so much with that information. The cutting edge technologies, so this is an experiment where we don't just stain for one thing, we don't just stain for T cells, we stain for 32 things at a time. And so we processed these samples using what's called multiplex ion beam imaging, which is a really long way of saying we threw 32 markers on there and tried to take pictures of it at one time. And so I know that this looks like a whole bunch of pixelated stuff, but what I'm circling here with all of these analyses and these pipelines that we've developed with collaboration through the very talented informatics team at the Parker Institute, is now we can take that biopsy from that patient and we can see a T cell, which is pointed on the top there. It's kind of outlined in like a bluish green. And we can also stain for the melanoma cells, which are outlined in orange. And what you're seeing there is a T cell actively dividing, actively seeing a tumor cell, and actively killing it. I know it's slightly underwhelming as a picture, but all that to say, we have come so far with these technologies that our capability to quantify these things, generate the appropriate comparisons, identify the appropriate statistics so that we can confidently say from a scientific angle, this is the therapy that is working for these patients. And that's what allows us to take the next step and move forward with the next wave of clinical trial design. So just in summary, immunotherapy fundamentally can build a bigger offense. While we have to keep working and developing, optimizing the therapies that are directly killing the tumor cells, we also have to think about how we can more specifically target them from the immunological angle. Patients with melanoma resistance to standard of care therapy, 29% of that 59% can benefit from this combination immunotherapy in the second line setting. And so all that to say, if we can identify these patients, the earlier we can identify that this is what's going to happen, the sooner we can intercept and hopefully give them a better outcome. And from my perspective, the biopsy analysis reveals the successful immune cell activation and T cell infiltration. And the fact that we have these cutting edge technologies and experimental assays to work with these clinical biopsies to drive the translational medicine is what will allow us to strategically go into the next wave of clinical trial design. I just wanted to touch on, as I introduced, I'm a postdoctoral fellow. This is an incredibly rare opportunity. What a postdoc means is I completed my graduate training, I completed my PhD, so I have to inform my family on every holiday, no, I'm not in school anymore. but I'm still training. But really, I'm trying to complete the appropriate steps, jump through the right hoops to start my own lab. And really, to me, that means training the next generation of cancer research. I get to work with a lot of very talented young individuals that are excited and motivated. And being a part of that is so, so important. But, I think back, and with any goal, you're thinking about your plan, and there's the finish line. You know what you're trying to accomplish. And for me, it was, I want to be a scientist. And the goal has become not that I want to be a scientist. I guess I am a scientist. But it's that I want to do good science. Obviously, this whole plan thing isn't exactly how it works out. It's a little bit more complicated than that. But the thing that those boats, those zip lines, the ladder, what else is in that picture? All of those things come down to the fundamental steps of my training that have required continuously applying to various fellowships and grants. And over the course of my postdoctoral training, I was first introduced to the V Foundation through the Gil Nickel Melanoma Research Fellowship, through the Johnson Comprehensive Cancer Center, and now I'm a part of the Bridge Fellowship. And what this mechanism does is it allows me to identify the appropriate career development that I need to continue to do good science. I also get to transition to hope to become, I'm an aspiring leader in this field. And so I get to emulate, hopefully, the mentors and leaders that have really set that foundation. And then from the type of research that I do, the fellowships, the type of research that they're funding is really advancing not just the information, but the cutting edge technology that is also being translated and utilized across all of the biomedical disciplines. And so with the ultimate goal of not just curing cancer, but preventing it and treating it and understanding it in the best way we can. And my favorite slide of all, when it comes to the acknowledgements, is that none of this is without the incredible leadership of the mentors that I have, the collaborators, and that spans biotech, pharma, nonprofit, and I'm incredibly grateful to stand up here today and thank all of you for your contributions directly to that and my training. So, thank you.
Thank you, Dr. Campbell. All right, please join me in welcoming Dr. Laquita Jones. Dr. Jones is an assistant professor of pediatrics at the University of Cincinnati College of Medicine. She's in the Division of Oncology. She works in cancer and blood diseases at the Institute at the Cincinnati Children's Hospital Medical Center. Please join me in welcoming Dr. Jones.
Thank you. Thank you so much to the V Foundation for this opportunity to be here today. Thank you for the invitation, Dr. Greer. I really appreciate this opportunity. This is a phenomenal opportunity. So I hope to enlighten you guys a little bit today about childhood cancer as it relates to leukemia, which is what I study. So the title of my talk today is A Foundation of Hope, Understanding Leukemia's Ability to Outsmart Targeted Therapy. So as mentioned, I'm Laquita Jones. I'm an assistant professor in oncology at Cincinnati Children's Hospital. Just a quick plug for Cincinnati Children's. It's actually ranked #1 as a childhood cancer organization and then also as a pediatric institution. So that's pretty cool. But I'm really excited to be here to share with you just a little bit of work that's being conducted in my lab. I want to start out with a little bit of background on leukemia. So I study AML, or acute myeloid leukemia. This is actually a blood cancer. So I'm guessing that most people in this room have heard of leukemia. I did want to start out with a quick question before I go into the background, actually, that I forgot to do. If by show of hand, can you guys raise your hand if you've either been impacted directly yourselves by cancer or if you know someone personally who's had cancer? Yeah. I figured it would pretty much be unanimous in the room. So for me, before I go into the background, I actually wanted to mention that my earliest memory of cancer, it goes back to me being a little girl, maybe like 4 to 6 years old. I can't remember the exact age, but my mom's best friend actually had breast cancer, and she died of it when I was 4 to 6 years old. And it's pretty interesting because I was reflecting on this at 2 a.m. when I was trying to sleep. And I'm still on Ohio time, my body is at least. And I was just thinking about the fact that at that time, my mom was my age. You know, she was in her 30s, and her best friend had died of breast cancer, which is pretty horrible, right? But in addition to that, years later, as a teenager, my mom's other best friend actually died of leukemia when I was 16 years old. And at the time, I didn't know that I wanted to be an oncologist, but I did know that I wanted to go into medicine. I've always actually wanted to be a doctor. If you would have asked me in first grade, what did you want to be, Laquita? I would say, oh, I want to be a doctor, a lawyer, or a teacher. And the doctor thing stuck, so here we are today. So in terms of AML, so acute myeloid leukemia, that's what my laboratory focuses on. This is a blood cancer. So we hear the term leukemia all the time, but this is actually a blood cancer. It's caused by abnormal growth and differentiation of immature blood cells. So what does that mean? Just to translate that a little bit more, so inside of our bones, there's this sort of factory known as the bone marrow. The bone marrow is responsible for producing blood cells. Now, those blood cells are supposed to go through this process of developing, maturing. So they start out as like baby blood cells, and they're supposed to mature and ultimately fall into their buckets or categories of what they're going to do. So it can be a T cell, like Dr. Richardson just mentioned. I think I got your last name right. It can be a T cell, it can be a red blood cell, a platelet, etc. But essentially what happens is when these cells don't properly mature and differentiate, they can sometimes start to grow out of control. So leukemia essentially happens when, this type of leukemia happens when you have those immature cells that are growing out of control. Now, in terms of how this shows up in our patients, because as mentioned, I'm a physician scientist, so that means that I actually am a clinician by trade, but I actually also have a lab, which I'll talk about my journey in a moment. But in terms of the clinical manifestations, it's related to the fact that those leukemia cells actually crowd out the bone marrow. So inside of our cells, inside of our bone marrow where the factory is, those immature cells kind of take over. So they crowd out the normal blood cells. So that results in patients coming in with essentially signs and symptoms of low blood cells. So they can have infection as a result of having low white blood cell counts. They can have symptoms of anemia. I think a lot of us have heard of that before, right? So you can have like fatigue, being really tired, having headaches, that kind of thing. In addition, they can have symptoms of low platelet counts. Now, a lot of people don't really know what platelets are, but platelets are actually important, and they help you to stop bleeding. So say if you were to nick your finger or something today, like on a piece of paper, and you stop bleeding within a few seconds or within a minute, that's actually due in part to your platelets functioning properly. So patients who come in with leukemia can have symptoms of bleeding like bruising or easy bruising or bleeding, such as nosebleeds, bleeding from the gums, that kind of thing. Now, in terms of survival as it relates to childhood leukemia, or AML in this case, is actually improved over time. So you can see here, if you focus in on the blue bars, I don't really have the ability to show my pointer very well, but if you just focus on the blue bars, you can see here from 1987 to 2012, there's been a significant improvement in the survival of pediatric AML, such that in the 90s, actually, the survival was about 50%. It's gotten up to about 70% now, and this is due to the fact that we can intensify our chemotherapy, meaning that we can give really strong medications that work really well in children. This is actually pretty different for adults, which I'll talk about in a moment. But in addition to that, we understand more about the leukemia, meaning that we can better risk stratify. So we can determine, okay, when a patient comes in with a leukemia, can we say that they have a relatively lower risk leukemia or a higher risk leukemia, and change the therapy based on that? In addition, if patients do unfortunately relapse, we have better options now. And importantly, we have better supportive care. So when we give chemotherapy, we know that chemotherapy can be pretty nasty, right? Patients are often in the hospital, they may need blood transfusions, things like that. They're at risk for infection. And we have better opportunities now to give them supportive care. So that means that we can safely give them blood transfusions. We can give them medication to prevent infections, which happens a lot as a result of the medications that we give them because their blood counts are so low. So we're essentially suppressing their immune system, but we can get them through this more effectively, in addition to nutritional support and things like that. So all things considered, survival has improved over time because we can give better medications, we can support them through it, and if they do relapse, we have better therapies, at least for children. So starting out with a case, this is actually a patient that I took care of when I was a fellow. So like Dr. Richardson, I did a clinical fellowship actually, because as I mentioned, I'm a clinician by trade, so I went to medical school. Then I did my residency training in pediatrics, and then a fellowship, which is 3 years in pediatric hematology and oncology. So that was the time that I trained as a subspecialist. So one of my first patients actually was a 24-year-old female, because we do sometimes see young adults. If they have a cancer that's more like a childhood cancer, we will treat them, of course. We'll treat anyone, obviously, but we're equipped to treat people who have childhood cancers in pediatric oncology. So she came in, she had sudden chest pain, shortness of breath, she had leg pain in both of her legs, she had a large bruise. Remember, I mentioned that patients who have low platelet counts can have bruising, and she had swelling of her left leg. Now, when they checked her blood counts in the emergency department, because she actually came in through the ER, all of her blood counts were low. So her white blood cell count was low, her hemoglobin was low, so were her platelets. Spoiler, we actually could see the leukemia cells floating around in her bloodstream, so we knew going into the bone marrow procedure that she did have leukemia, but we had to do the bone marrow in order to get additional information to understand more about this specific type of leukemia. She was ultimately diagnosed with AML, and this is actually one of the things that led to me studying AML in my laboratory. So, as I mentioned, I'm a clinician, but during my fellowship, I joined a laboratory as a second-year fellow, and that was in 2016, actually. I joined my mentor's lab because I wanted to study AML, and the reason why I wanted to study AML was that one of my first patients, who wasn't this one that I mentioned, but it was actually a teenager who was 15, and unfortunately, she did die of AML. And it was during my first year of clinical fellowship that I recognized that there's still a lot of work to be done in this space. Yes, there's the BALO category of patients where there's a greater than 90% cure rate and that kind of thing. And that's not to say that children don't die of ALL, because they do, unfortunately. But I saw a striking difference with the AML patients. In fact, like I said, my first patient, 15 years old, I adored her, so did her family, of course. She died of AML. And I recognized that there was a great need in this space. So I decided to join a laboratory, which was not my plan when I went into fellowship. I knew I was going to do research of some sort, but I thought I would be a clinical researcher, not a basic scientist. I hadn't touched a pipette since college, and I wasn't planning to, but then I decided to join a lab. So here we are now 7 years later, and I have my own laboratory, which I'll talk about momentarily. So my laboratory is focused on understanding more about novel resistance mechanisms to targeted therapies in AML. And I think this is a really important topic because, you know, I tend to be somewhat of an optimist, but I think if you talk to some people would say that with targeted therapies, that it's inevitable that resistance would happen. I don't personally subscribe to that thought process just because, like I said, I'm pretty optimistic. You know, and I think we've made a lot of advances, but nevertheless, it is a really important topic, right? That's actually the point of this whole weekend, because we know that targeted therapies do lead to resistance frequently, unfortunately. So as it relates to survival of pediatric AML, it has reached 70%, but it's also plateaued, unfortunately. So it reached 70% in the late 90s, early 2000s, and this again is due to the fact that we can better risk adapt our therapy, meaning that we can understand more about what is the risk of the leukemia, what medications do we need to give in order to treat this patient more effectively, in addition to the fact that, as I mentioned before, we have better supportive care. Now, in terms of how we treat AML, it can include, and it does frequently include at this point in time, at least in the pediatric space, chemotherapy. But as I mentioned already, and as you know, chemotherapy has a lot of nasty side effects. We also know that chemotherapy can later lead to secondary cancers, unfortunately. But in addition to chemotherapy, there are targeted therapies available, which we utilize. Actually, one of those targeted therapies I'll talk about momentarily, which is the focus of my lab. So these targeted therapies are nice because they target the specific abnormality within the cancer cell, right? They tend to have less side effects, which is nice, but they also have downsides, unfortunately. Treatment for AML can also involve radiation therapy. It's not frequent, but it does happen sometimes that we use radiation therapy. And then importantly, AML therapy does sometimes involve stem cell transplantation. I would say this is a more common thing in the adult setting. In other words, bone marrow transplant. So essentially the idea behind a bone marrow transplant is that you essentially give really high dose chemotherapy, you wipe out those kind of workers in the factory of the bone marrow, and you replace it with new workers from a donor. So that means that can be a sibling, it can be a parent sometimes, it's usually a stranger, but this actually highlights the point that it's really important that we have diversity within our registry for the bone marrow registries, because part of the problem with bone marrow transplantation is that people of color sometimes don't have access to a donor because they may need a donor that is, you know, perhaps of a different descent. And a lot of our bone marrow registries actually are from people who are European in ethnicity. So in terms of survival, again, from 2011 to 2021, there's been minimal change and improvement in treatment outcomes. And the reason is that I know, this is based on literature actually, we've reached a plateau because chemotherapy can only do so much. And that highlights the fact that we do need targeted therapies or immunotherapies, et cetera, to kind of get beyond this plateau of 70%. And I will say that the survival in adults is strikingly different. It's actually the opposite. So you think maybe 30% of children who have AML won't survive. Well, in fact, 30% of adults who have AML do, and 70% don't, unfortunately. So that being said, there's a lot of work to do in this space, both for pediatrics and for children. As it relates to targeted therapies, they're being utilized more frequently to treat AML. Prior to the 2010s, there was only minimal change in the standard of care to AML therapy, meaning that we frequently use chemotherapy, but not really anything else. Not many targeted therapies were being utilized during that time frame. Since the latter 2010s, there have been more clinical trials utilizing targeted therapies in AML. In fact, there was actually a study that was published in 2023 stating that over 30 targeted therapies are currently being utilized in clinical trials or clinical settings to treat AML, which is fantastic. However, as mentioned, there are downsides to targeted therapies, right? We know that patients can acquire resistance, or they can go into remission with targeted therapies and then relapse with the type of cancer that's, so to speak, outsmarted the targeted therapy, unfortunately. So my laboratory studies FLT3. So what is that? FLT3, or FMS-like tyrosine kinase 3. This is a protein that's present on the cell surface of blood cells. So this is a protein that's actually present in normal blood cells. So it's present, it basically helps to promote growth and division of those blood cells, and typically FLT3 actually has a built-in safeguard such that the protein itself can actually turn off. So it has this built-in auto-inhibitory brake, so to speak. Unfortunately, a subset of leukemia patients have cancers that can hijack that functionality of FLT3 such that the protein is always activated, is always turning on, is always promoting division, that sort of thing. Now, FLT3 has actually been targeted over time with targeted therapies, so FLT3 inhibitors, and that's what my laboratory focuses on. We're interested in this FLT3 F691L mutation, which is a fancy sort of label, so to speak. Basically, this is just a type of mutation that happens after targeted therapy. So after FLT3 inhibitor therapy, it only ever happens in that setting. When it happens, this can be challenging to eradicate. So these patients can do poorly when they develop this resistance mutation, unfortunately. Now, I don't think that we fully understand how this mutation works, which is why my laboratory is focused on it. This F691L mutation is known as a gatekeeper, meaning that it should theoretically prevent these inhibitors from binding to FLT3 and prevent them from inhibiting the functionality of FLT3. However, I think there's actually more to the story, because we have data to say that this doesn't just function to prevent binding of these inhibitors, it actually does other things, which is the nature of the research that's funded by the V Foundation. So again, my laboratory is trying to understand the exact mechanism of resistance related to this mutation. We're trying to do this through a variety of different approaches, including genetic and therapeutic manipulation of different cell lines. We use human cell lines frequently. So in terms of therapeutic manipulation, of course, we use FLT3 inhibitors, but in addition to that, we use other novel compounds that we have created to try to target this specific mutant, in addition to genetic manipulation, whereby we can disrupt the expression of this mutant form of this protein to target these leukemia cells. We want to really understand how are the leukemia cells being rewired to promote this resistance. How do they rewire themselves? How do they partner with different proteins so that they can then say, oh, you know, we don't care about the fact that this FLT3 inhibitor is present. We can still continue to thrive in this patient and ultimately cause poor outcomes. So the ultimate goal of my research is to have a greater understanding of the resistance mechanism of FLT3 F691L and to identify new therapies. So not only do we want to understand how this happens, we want to understand, are there new proteins that we can target? For example, could you use a combination therapy with a FLT3 inhibitor plus another targeted therapy to overcome this resistance or to prevent it from happening in the first place? In my mind, the best thing, the ultimate goal actually is to prevent it from happening in the first place. So, in summary, AML survival has improved over time, but there is still work to be done, unfortunately. There's work to be done in the pediatric space, and there's work to be done in the adult space. AML is a blood cancer, as I described earlier. It's the second most common acute leukemia in children, meaning that it's a leukemia that involves those immature cells that I mentioned before. But it's actually more common in adults. So to give you numbers, in children, there are about 500 new diagnoses of AML in children per year, and about 18 to 20,000 adults per year. So it's a lot more common in adults. Survival in children has plateaued at about 70%, and survival unfortunately remains really low in adults, and I think a lot of that has to do with the fact that adults often cannot tolerate that really intensive chemotherapy that I talked about. You know, adults may come in, they may have kidney disease or heart disease, congestive heart failure, something like that. That precludes them from getting chemotherapy that will be really intensive. And while chemotherapies are effective, targeted therapies are needed to improve outcomes in our patients, as we know, and to limit side effects. However, we know that targeted therapies have their own downsides with higher rates of resistance and relapse. So the V Foundation is helping to fund my work in my laboratory at a pivotal time in my career, because the reality is I'm still early on in my career, and these types of awards, so I have a V Scholar Award, which I'm really thankful for. Thank you so much. But this is a really important time in my career. Oh, thank you. Thank you. I wasn't expecting that. Thank you. But I have a V Scholar Award, and this is an important time of my career because I'm still a junior investigator, and I do have a laboratory, but I'm working towards independence. But awards like the V Scholar Award serve as sort of the way that I think about it is like a launching pad. So it's a really important, pivotal award early on. I really love the strategy that they have of funding early career investigators, because the reality is, number one, it's highly competitive, so in case you were wondering, it is. It has a reputation, which I think is phenomenal, because to me that highlights the rigor and the excellence that they're looking for in terms of the productivity of the research, right? The quality. But in addition to that, it's really a nice launching pad, because then it opens the door for other grants. Because when you're applying for grants, one of the things that the reviewers sometimes look for is that you've gotten some grant funding previously. So this is a really important award to me. And actually, I think that this work that I'm doing could not only apply to AML, but also to other cancers, because in fact, these targeted therapies and these gatekeeper mutations have been described in other cancer types. So I'm hoping that if we can really understand how these quote unquote gatekeeper mutations work, then we could apply this more broadly to other cancers that are not just leukemia. This is my acknowledgement slide. I want to acknowledge my laboratory, my collaborators, my career development committee, my funding sources, including the V Foundation, and I did get a career development award from the National Cancer Institute, which is huge. Since I've gotten the V Scholar Award again, this is highlighting the fact that this is a launching pad. So thank you so much for this opportunity. It's an honor to be here today.
Thank you, Dr. Jones. All right. Please join me in welcoming Dr. Jae Hook Choi. Dr. Choi is a V Foundation scholar. He also is an NIH New Innovator. He is the Jack W. Graffin Associate Professor and Director of Precision Immunotherapy, co-leader of the Immunology Technology Working Group at the Robert Lurie Cancer Center at the Feinberg School of Medicine at Northwestern in Chicago. Join me in welcoming Dr. Choi.
So I just wanted to say thank you all for inviting me, and it's just a really pleasure to be here. And just inspired by the previous talks, I want to talk a little bit about my journey and how you're just a critical part of this. So I have to say, as I'm an immigrant kid, you know, my parents came over on the 4th of July many, many years ago. And my parents had a little store in West Philly. And my mom always asked me to make an impact. We're in a new country where you can do whatever you want, and I just think that you should try to make an impact in your life. And so inspired by that, when I went to college, I decided I wanted to do science. And I just wanted to share that I did this screen. It was a very complicated scientific experiment that I was very proud of, that took over 2.5 years. And I remember very specifically that I actually presented my research to an interviewer at Cornell Medical School. And I remember very clearly he told me that I got it all wrong. And I had done this complete genome-wide screen, and I had found a gene that was called the prostate apoptosis response gene 4. And I was telling him that we're going to find everything that this gene does, and our hope is to improve the lives of patients with prostate cancer. And the reason why he said we got it all wrong was he was like, you should start with prostate cancer. and then go backwards, find the genes that are relevant for those cancers, and then you can do your fancy assays. Otherwise, we'll never improve health. So I have to say is they couldn't have paid me enough to go to that medical school because it was an extremely challenging time. But I have to say very humbly that this one interviewer's advice, I actually remember his name very well, has inspired me. And I think that we have come to a time where we can do exactly what he had mandated, which was, let's study patients, figure out what makes each of them tick, and this is the only true way by which we can develop therapies. So I just wanted to end by saying, in this part before I continue, is that, I'm also a physician scientist. I'm actually a dermatologist by training. I just found out that Bill Nelson and I are cousins, actually, through our scientific tree. But, just before I came here, I didn't have time to put a picture, but I saw one of my patients who actually failed the therapy that I'm going to talk about. And I went to go see him as he was receiving the second-line therapy, and I just made him a promise. I said, I'm sorry you're going through this, but we got to do better, and I will do better. And so I just want to say is that we are always doing our research for our patients, and so that really inspires me to do what we do and to talk about what we're going to talk about today. The other disclaimer I have to say is that some of you might find my slide deck very simple. And if you think this is so, just know that I had originally given a talk like this to Congress about 5 years ago. So every time I tried to put something together, my Northwestern team just said, lower the level, lower the level. Okay, so that's all the disclaimers you'll get. So I'm actually a dermatologist, I said. I study the skin, and one of the reasons I went into it is because I really had a heart for patients with skin cancer. And so one of the things I wanted to say is that the skin has many different cell types in it, as you can see in this slide deck. And they include keratinocytes, which lead to basal cell carcinomas and squamous cell carcinomas. You've heard about the melanomas, which you just heard about. This is probably the most famous skin cancer. But there are also other cell types in the skin, including T cells, B cells, and Merkel cells. And all of these can become cancer as well. So my particular V Foundation Translational Research Grant actually focuses on the Merkel cells, which is an uncommon cell type in an uncommon cancer. But we think this is very important for our patients, but also for cancer at large. So the reason why we think this is so important is because this Merkel cell carcinoma is such an unusual cancer. It can be caused by two things. So about 80% of patients actually develop cancer because they have a virus on their skin that goes into the genome of the cell causing the cancer. And then about 20% of patients actually have just too much UV light, too much sunlight that causes all these mutations, and this also causes the cancer. And we know these two processes are sort of interacting with one another because in Australia, the ratios are flipped. So about 80% of people in Australia actually have UV-induced cancers or sunlight-induced cancers, and only 20% are caused by viruses. The reason why this is so important is because for so long, as you've heard, actually this was the cancer that you didn't want to get. There were absolutely no therapies that actually improved survival or caused any durable responses. But what we have learned in the last few years is that this cancer is incredibly sensitive and visible to the immune system. And so what happens is, both by having a virus in the DNA or the genome, or by having mutations in the DNA, the cancer cell shows that it is different from all the other cells in your skin and in your body. And this is a signal, which I think I show by this red star, which shows that I'm different and you should get rid of me. And so what happens is, the T cells come around and they want to kill the tumor cell. But the problem is the tumor cell is too savvy, and they've developed resistance mechanisms, as you've just heard about from Dr. Campbell, which is this PD-L1 ligand is one example. It's a molecular break that prevents the T cell from recognizing and killing the tumor cell. So what we have been interested in is therapies that actually disrupt this defense mechanism for cancer that enables the T cell to recognize the tumor cell and actually complete its mission of killing the tumor cell. So I wanted to highlight this slide. I think you've just seen a couple examples of this. This is the survival curve for patients with Merkel cell carcinoma with immunotherapy. And so when this was published, it was actually an amazing accomplishment. We had no treatments that were curative or even effective for patients with late-stage disease. Now you can see that there are actually about 30 to 50% of patients are cured. And so we were incredibly humbled, but also incredibly energized as physicians by seeing this. But I want to highlight the focus of our lab, which is what we see in yellow. Our goal is really to help the people who don't respond to therapy. About 50% of patients have primary resistance, which means they do not respond to the immunotherapy, despite the fact that if you look at the cells, they have every reason to be visible to the immune system and should be cleared by the immune system and these therapies. So what our lab does is, I think, getting back to my interview at Cornell, is we've done exactly what the interview has mandated. And our goal is really to understand, why are people different? And so in gray, you'll see these are all patients diagnosed with the same cancer. And what we have been focusing on is using the molecular tools in our lab to identify what are the major drivers for each person's cancer. And that's the colors that you see. So we know that even though they look the same in person or through any medical test, that there are molecular differences that make some patients blue, red, green, or black. And the focus of the V Translational Research Grant that I have is to identify those colors, but also to try to convert this to the next step. So the next step is not just finding the differences that make each patient different from one another, but also to make sure that that patient receives the right therapy. So our goal is to take the patients who are blue, red, green, yellow, and make sure they get the medicine specifically for them. And so how do we do this, and what have we been funded to do? What we actually do is a very, what I call the bedside to bench to bedside approach, which is that we take patients, and then when they undergo surgery or a biopsy, what we do is we take the patient samples, and then we take the tumors, as you see here, and then we do a number of really complicated analyses. So I just show here a map that was alluded to earlier by Dr. Campbell. This is a single cell spatial localization map, where you can see every single cell in the tumor biopsy, identify with really high precision what it is, and with molecular data now, you can figure out what it's doing. And so we now have this single cell or single molecule map, and we've done this for over 100 patients, and we've done over 500,000 cells, where we can identify all the tumor cells, all the immune cells, and how they're interacting with each other for each of the patients. And then utilizing, the really bright people in our group, we do machine learning type approaches. So these are the computational approaches that are really required to handle this kind of data. So as an example, I think we have about 100 terabytes of data between all of the different things that we've done. And this requires cloud computing, a number of really expert computational biologists, as well as the biostatisticians, to help us think of even new ways to look at this data, which is kind of first of its kind. And so we utilize these, and this is the sort of simple colors that you had seen before for the patients, that we can actually use the tumor cells themselves, as well as the molecules and cells in the tumor, to understand how each person is different, and to identify the groups that respond or don't respond to current therapies. So the way that our proposal is working, and I'm really glad, because I actually thought I was in my second year, but I'm transitioning to my third year of a third-year grant. And I just want to say that we have, I think, done almost everything that we set out to do, which is actually kind of rare, I think, for a grant, for many reasons, which I'll discuss later. But our goal is really to not only identify who responds and who doesn't respond and what color they are, but we've actually identified a number of targets that are really critical for the next stage. Because, all of our patients are interested in knowing if they'll respond or no respond, but what they're most interested in is, are there therapies for the ones who don't respond. And we've been very excited to find that we have identified many of these targets that are lining up for the next range of clinical trials that we're trying to perform on this patient population. So that's what we're going to show here. And so I can definitely talk about this individually, and I'll show you some of these examples, but we're actually really excited by the fact that we're on we're going to start ending our second year of this award and starting our third. We're already trying to launch a multi-institutional trial based on our work, and then we have about 3 or 4 targets that we want to go to in the near future. So I just wanted to highlight, this is Zachary Reinstein. He's a MD PhD student in my lab, and he's worked very hard on this process of trying to figure out new ways to improve therapies. If you would just allow me the quick sidebar, we've just heard about how long the training is, and that's why this pipeline is so critical, and the V Foundation plays such a critical role. I told him, and it's sometimes hard for him to imagine, but from the time he started medical school to the time he finishes his postdoctoral fellowship, a kindergartner will graduate high school and be midway through their college career. And so I just want to say is that, I'm so fortunate to have this brilliant genius in my lab who can handle these terabytes of data, but we have to protect him. His goal is he wants to be an oncologist and figure out new ways to cure cancer. We have to help him along the process, which is part of what the V Foundation is really critical to do. So in our experience, what we have found is that there's generally 2 types of patients. We have the patients on the left, which favor response, and patients on the right, which favor resistance. And so on the left, we found a number of different cells, immune cells in the tumor microenvironment. Some of these are already known in other cancers like melanoma. We've identified molecules that prevent them from killing the cancer completely and are designing clinical trials to sort of overcome these barriers. We've also identified actually non-conventional T cells. These are these yellow cells, which are not the T cells you heard about earlier, but there are other types of immune cells that we really know very little about, except for the fact that they tend to be very common in some patients' cancers. We think these cells are actually doing 80% of the work of trying to clear the tumor cells. We just need to develop new therapies to be able to help them to complete their mission of actually curing the cancer. And then we've also identified molecules that are produced in patients who don't respond that seem to be suppressing the immune system. So we're taking this 3-pronged approach of identifying novel targets and trying to launch clinical trials related to our work to be able to offer our patients who don't respond to standard of care medications. And as a plug for the rare cancers, this is a rare cancer, which is, I think, defined by 40,000 cases or fewer in the US. Altogether, rare cancers make about 27% of cancers in the US. But just as was discussed on a few nights ago, we think that the lessons we've learned from this model cancer, we can actually exploit, and we're actually also gearing up to potentially do clinical trials in other cancers that are more common, like lung cancer and potentially pancreatic cancer based on our work. So to summarize basically where we've been and where we're going as we end this 3-year award, we are trying to implement a network where we can study patients. These are tumors extracted. And if they have the favorable characteristics, our goal is to get them as quickly as possible to the therapies that we know will work. But the other arm, which I consider very important, as I said, I really made my promise to my patients that we'll do better, is that we really want to identify the patients who don't do well. And so don't waste their time going through standard of care, treatments, and instead go to clinical trials that are actually designed specifically to help them. So what we're looking at are antibody-based therapies that we can leverage to try to do clinical trials now. And then in a new area that's exciting, we're also working on engineering cell types that can actually be utilized to also help along this process. But this is all sort of may seem like science fiction to me, but actually it's something that we could potentially do in the next 5 to 10 years, and with our new clinical trial, hopefully within a few months. So the reason I want to talk about the V Foundation, what it means, is because I told you when I applied to medical school, I didn't think you could start research from the patient. I thought it had to start from a molecule. And at that time, just looking at, the hint of a genetic disease to identifying the gene that causes it could take decades. But within 3 years of this grant, we took what we had very little knowledge about these cancers, and we're trying to launch this clinical trial in multi-institutions now. And so I think there's every reason for hope, and we just think it's a wonderful time. And it's kind of like, it's the perfect time to try to encourage all the factors that will lead to cancer cures. And so I think this is my last slide. And so you might ask me, what does it take to cure cancer? I said the first thing is you need really bright people who want to work on this problem. So Zachary is my MD PhD student who's working on this. This grant has enabled us to do almost anything that he imagines, actually. And so I never have to tell him we can't afford that. And so it's an incredible experience to unleash this incredibly talented person on this very important problem. The second thing I think you would say is if someone asked you, do you need bold ideas to cure cancer or conservative ideas to cure cancer? I would say you need bold ideas to cure cancer. So that's number 2. And then number 3, we just talked about this, but technology has advanced so much that we're able to do things we never would have imagined. And so I just want to end this with a comparison. So most of us in research actually get a lot of our money from the NIH. And it's very difficult to do this, but the reason why it's so hard to cure cancer this way is the NIH is extremely competitive, so they're extremely conservative. And in general, they don't fund ideas that you haven't done yet. And obviously, you want to have people who can dream of doing something and fund those ideas. Secondly, but from the time you start to put in the first application to getting an award, it can take over 2 years, actually. So you just have to know it's so slow. And then generally, they fund incremental science because of the conservative nature. What I feel really indebted to for the V Foundation was it's extremely fast. When you have an award that they like, they fund you. They give you enough resources to fuel the creativity of your scientists, as well as fuel the bold ideas that they have, as well as get the technologies you need. I didn't talk about this, but one of the things that's great about the V Foundation is they've also facilitated multi-institutional efforts by us to link people together with minimal bureaucracy, which has made it very easy for us to quickly translate our work. So in summary, I just think that this is an incredibly important organization, really devoted to solving an incredibly important problem. And I think it's an incredibly opportune time to actually expedite us towards a cure for cancer. And just thank you all for all of your support. Thank you.
Thank you. Those were wonderful presentations. And now to probably our favorite part. I know you have questions for Drs. Davidson, Dr. Choi, Dr. Campbell, Dr. Jones. You know our SAC is here, so please, if you have a question, raise your hand and we will bring a mic to you and you may ask a question to me, to them. Please, join us.
Thank you. I have more of a comment than a question. This is my first V Foundation event, but I'm a 31-year pediatric surgeon, and I just have had a long personal connection to fighting cancer. I was recently at the American College of Surgery meeting, and at breakfast, some residents sat down at the table with me, and I said, gosh, I'm a 31-year pediatric surgeon, and I still love what I do every day. And they said, what's your secret? And without hesitation, I said, take your care personally. Get to know your patients. Connect with them. In pediatrics, it's easy to treat children, my patients, as my children. And to hear these personalized approaches and now having a cancer treatment that needs to be personalized for people's genetics, as well as the molecular biology of the tumor, that the personal approach, both for our scientists and our surgeons, as well as for our patients, is the way forward.
Thank you. Thank you. Yes, there's a question in the middle. Thank you, Megan.
I just was curious, any of you can answer this, but how is this information communicated internationally as the research is unfolding? It seems that there's been a lot of really innovative work done with wonderful outcomes. Is this work shared widely and for anybody to access?
Would one of my speakers or one of my SAC members feel strongly? Katie, Dr. Campbell.
I would say for the scientific community, we focus on publication and, various types of media have made that very much accessible. I would highlight one of the roles of the V Foundation, the nonprofits in these consortia, and working with the SWOG consortia. I've come to recognize this, that getting and communicating this information not just to the research centers but also to those community centers has been instrumental in getting that information out there. And so I think we need to continue to do a better job of exactly that. But I will say that is stimulating these conversations at events like this, but also figuring out how to use the broader media. And I think ESPN is also working on that, and we greatly appreciate it. I think we need to constantly do better. And so I think it's rare for me to consider this type of opportunity because the way that I'm talking about my science now is not necessarily how I would speak to my boss. And so figuring out how to continue to perpetuate these opportunities for the scientists as well will only continue to expand upon that.
Thank you. Yes, there's a question in the second row. Yes.
The United States as a country spends more on medical care per person, both individually, but also the P&L of the United States of America. It seems to me when we get past the emotional part that challenges us all and these very talented people, and those costs obviously have lawyers who are defensive in medicine, which causes us also some difficulty. It seems there's a lot of trial and error. It seems there's a lot of silos instead of a horizontal sharing. We all know situations where labs on the East Coast are fighting with labs on the right coast, the left coast, to see who gets credit for things. How do you take a horizontal approach to a problem and bring an amalgam of people challenging it and get these costs down? It takes 8 years and $3 billion to create a drug. We're headed off a cliff.
It's a great question. Let's see. Let me hand that to my colleague, Bob Bast. Dr. Bast, would you take that question?
That was a very good question. You know, it's amazing. I think you hit one of the issues right on, that this is an inefficient process. And it's got to get more efficient. You know, these days, only 1 in 8 of the drugs that is brought to the clinic for clinical trials, after often hundreds of millions of dollars of investment, is actually approved. And it's not because the FDA is being tough, it's because the drugs don't work. And that's because our preclinical studies in cells and in mice don't predict what happens in people. And that's why the message you heard earlier today about studying the patient and what's happening is so important. More investment needs to be made in better models to make this process more efficient. There are a number of social issues, certainly, and governmental issues that need to be addressed as well. But there are serious scientific issues where we need to put much more emphasis.
Thank you, Dr. Bass. There's a question in black. Let's see, near the back, yes.
Suzanne, I think it'd be interesting for the audience to hear how much funding each of the individuals that spoke today is receiving from the V Foundation.
Absolutely, I'll let them speak to that, and then Maybe I'll precede that by saying the V Foundation has recently changed our funding structure because these are current grantees. Can I answer the question in a different way? All right, so these are current grantees for the V Foundation, and we have increased our funding in recognition that our grants were too small. Our grants before 2000, so this is a bit of a plug maybe for Dr. Greer and the Scientific Advisory Committee. I joined the V Foundation in 2022, and we recognized that our grants were not big enough to do the science that our grantees needed to do. So we have 2 legacy awards for the V Foundation. They are the V Scholar Award that Dr. Jones spoke of, and the V Translational Grant that Dr. Joy has. When I joined the foundation, the V Scholar Award was $200,000, and the V Translational Grant was $600,000. And so we, in discussion with the Scientific Advisory Committee, decided last year that that simply wasn't enough. Science is extremely expensive. Just to keep up with inflation, the V Scholar Award needed to be $487,000. That's just to keep up with inflation. And that doesn't keep up the scientific inflation index, which is much higher than just the inflationary index that we put on the American dollar. So now, beginning in 2023, the V Scholar Grant is $600,000 over 3 years. The V Translational Grant is $800,000 over 4 years. And I believe that those awards are now appropriate. They are still small. Our grantees need additional dollars from other organizations, but they are appropriate to allow these researchers the launching pad to begin their careers. They are given at the point of, because they are high risk, not everything that these individuals are planning to do is going to work. And that really is okay. Because if just a piece of it works, that's going to be enough to help them get, as Dr. Choi says, some of those grants from the National Cancer Institute that will allow them to do some of the work that will be bigger grants, perhaps maybe, I don't want to say less innovative, but maybe bigger grants that will be structured and have in them studies that are able to show that these are absolutely necessary. They will have that V-funded data that is powered, that perhaps is less risky because it's already been funded, and you can show the National Institutes of Health that yes, this is going to work because the V has already invested in that high-risk, high-reward science. So I hope that answers your question. The grants that these individuals got were smaller, simply because at the time the V Foundation was funding the smaller grants. I hope tonight you'll dig deep to fund those $600,000 and $800,000 grants. Yes, there's a question here on the right.
So we're all, I've been involved with the V Foundation for many years, and we're all very excited about the V Scholar, the translational, because of what you said, Dr. Choi, about taking a bolder view of things. And I'm curious about the comment you made about the NIH, and I'm wondering if there's anything being done to try to transition the NIH to making bolder bets like we're doing here, because they have a lot more money than we do, and that would be great. Anything being done about that or can be done about it?
I'll pass that to Dr. Davidson.
I think that, first of all, remember the NIH is you and me, right? It's our tax dollars. So we're investing on behalf of the entire country, and you may want bold and innovative. There may be other taxpayers who want a little bit more conservative approach, so we need to balance that. And it is the workforce, it's the engine for what we do. But the NIH is thinking about this too. For example, they have a grant mechanism for the last decade or so, something called an R35 grant. And Olya, are you an R35? Yeah. So I'll introduce it, and Olya will tell you what it's done for her. So this is a grant that recognizes somebody who is a more seasoned investigator and says, you have been a success. You have some really cool ideas. We want 6 pages, was that how long it was, a 6-page outline from you of what you think you're going to do generally over the next 7 years with $1 million a year. And we're going to go with it. So Olya has one of those, and I would say that's what you're talking about, innovative, right? She didn't have to sit down and say aim 1, aim 2, aim 3. She said, this is what I've done, this is the problem, this is where I'm going. Olya.
Thank you, Nancy, for this plug. I'm actually just about to finish that award. And I have to tell you that I actually wrote about 4 aims, and the last one I said, this is my auspicious aim. Didn't even say this is going to work or not work. This is like, I'm going to try this. I made a comment the other day that we should, in our search for cure, also spend just as much time thinking about prevention. And so this was a cancer immunoprevention grant at the time when people were not really talking much about immunoprevention. And so they trusted me to figure out, how to go about that, not only in my own lab, but actually to get other people excited to be the advocate for that particular area of research, and the grant is from the Division of Cancer Prevention. So, but I also want to add that, and I have to say, we've done a lot with that grant. We've run clinical trials and published vaccinating people with the cancer vaccines, healthy people at risk for cancer. And today, and in terms of getting other people to move in that direction, I got an email today from the University of Pennsylvania, from the Basser Cancer that is a center, Basser Cancer Center, that works on BRCA1 and 2 risk and carriers. And they're announcing, we're excited to share an update about our efforts to intercept BRCA-related cancers. We have administered our cancer prevention vaccine to the first healthy individual with a BRCA mutation. That's it. And so it's a long road, but you need advocates, you need people to show that some of these things will work. So I'm very excited about that. But I also want to add one more thing, that I've been on study sections at the NIH, at NCI, and we are trying, even as reviewers, to strike a balance between something very high risk and sort of high risk and potentially high reward. So it's up to the reviewers, actually, not necessarily to the policies of the NCI. Just like we do here for the V Foundation, we pluck those ideas out, and give you these kinds of people.
And just very briefly, the NIH has several different mechanisms. There are a couple of grants that are smaller grants for which you have no preliminary data, just an idea. So there are sort of a series that unlike the R35s, the more you ask for, in general, the more data you have to bring to show that the idea has merit. But there are several different mechanisms. There are R03, R21, I could give you more details. There's another mechanism of working forming a company to both SBIR, to a small company working with the investigator as well to try to commercialize things more quickly. So it isn't completely stodgy.
It's a good combination with us.
Thank you, Dr. Lawrence. Yes, good combination. Yes, in the middle. Thank you.
So this is for Dr. Bast. I would like you to share your piece about why exactly what we're going to do tonight is so important and why it has to come from the public sector.
Thank you. Thank you for the question.
Absolutely. Actually, this gets into a political discussion. One of the things you got to know about cancer research is it's bipartisan. This is one of the few things everybody agrees ought to be done. But this year is a little bit different. With the crisis about the debt ceiling, the compromise that came out of that, I think as many in the room are aware, was that the federal budget's going to be absolutely flat. And despite that, you could still, by decreasing some things, increase cancer a bit. But when you put this in perspective, the federal budget's $1.7 trillion. The NCI gets $7 billion. That's 0.4% of the federal budget. And once you take out defense and Medicare and Social Security, everything else is discretionary. So you're fighting about so many worthy things that need to be funded. So I think the House has already made their recommendation. I think the Senate, when the break occurred for August, was still thinking about this. But it turns out that they wanted to give NCI $200 million less this year. And they're already about 20% behind because of inflation. So this would mean instead of 12% of the grants being funded, it would be 8%. And that just isn't acceptable. But this is why it's so important tonight, because you need to fill those gaps. And this is why the V Foundation is even more important than ever. We obviously need the National Cancer Institute to fund these things long-term after the really innovative ideas have been developed far enough to get long-term funding. But at least particularly for this year, what the investments that the V Foundation is going to make are even more important.
Thank you. Yes, there's a gentleman in the hat in the middle. Thank you.
It's a V Foundation hat.
It's the best hat.
I'm overwhelmed by the large number of variations of cancers, and I myself don't have a working definition of what makes a cancer. That's my preface. My question is, do we have a handle on all of the major forms of what we call cancer? And is there a commonality in identification of causes and treatments between these various cancers? We've heard a lot about individual cancers and what people are doing. So are there common building blocks to solving this problem?
It's a great question. Let me hand that. I was just thinking Dr. Shelley Earp, and he raised his hand, so great minds. I will say Dr. Earp and I saw each other in the gym this morning, so that's one thing we can all do.
Exercise prevents. The answer is yes and no. There are a set of genes. You have 18,000 genes. One of the amazing groups of genes are about 400 or 500. And those 400 or 500 actually go around and check your DNA so that you can get rid of mutations. You make 75 quadrillion base pairs in your body every second. So your body has evolved a system that keeps it from mutating. There's another set of genes, two other sets, ones that make our cells grow when they should, and ones that prevent the cells from growing. So the commonality in many, many cancers that we target with these targeted therapies are either the ones that get mutated that make it go fast, and the ones actually that are more difficult are the ones that get mutated, which are the brakes. So that's the commonality. However, there's differences in virtually every tumor type. So we heard about Merkel cell, we heard about AML, we heard about melanoma, and each one of those has some differences. You also saw some great technology, which is letting us delve down into what these differences are. So again, yes, there are certain commonalities in genes which are almost always mutated, but no, there are differences that we have to dig down and really personalize. Personalize in the tumor type, but also personalize in the individual patient.
Please, thank you, Dr. Earp.
So all of these amazing differences are once the cancer is already there. When we think about prevention, we would like to intercept, treat at the time when these mutations have not yet happened, these millions of different mutations. So for prevention, we're looking for the commonalities that you're asking about. So for example, I can tell you that over 70% of all pancreatic cancer starts with a mutation in one particular gene, KRAS. So if that's the first mutation, and if we know that's going to happen, then, a lot of people are working on vaccines against that particular mutation to kill the very first cell that mutates in that way, so that other mutations will not happen. So this heterogeneity is heterogeneity of an established tumor. But they all can start from a very specific, maybe single mutation. So I think for treatments, we have to be aware of all these variations. For prevention, we're looking for commonalities, and they're there.
Thank you. I think maybe from one of our biostatisticians, too, an answer would be, I'd be interested to hear from, I'm looking at two of you, so. All right, I'm going to go, yeah, absolutely.
So, hi, I'm Susan Hilsenbeck. One of the things that comes to mind, because I mostly work on, or I have most of my career worked on breast cancer and was contributing to clinical trials related to HER2 and the successes around HER2-positive breast cancer and that we're now able to treat. Now, that was developed in breast cancer. Most of the drugs were developed in breast cancer, but we use those same drugs for other cancers as well that express the HER2 amplicon and express the HER2 protein on their cell surface. Not all cancers that express that protein respond to these drugs, but quite a few do. And Dr. Davidson can probably comment better than me on the dichotomy between that. So there definitely are commonalities where we learn about drugs that work in maybe a more common cancer and then can apply it to other cancers as well.
Thank you, Dr. Hilsenbeck. Okay, there's a question in the middle.
Sorry to ask the second question, but Dr. Bast and Dr. Choi. Dr. Choi said some magic words. We've got less money and a bigger problem. You mentioned innovation. Innovation solves problems, not just constantly throwing money at things. You mentioned ML, machine learning, for data, huge data, more data, the cloud, where you don't have it in a bucket somewhere, everybody can tether off the same data. You mentioned computing, which means supercomputers. You mentioned biostatistics. You're the generation now that takes it to the next level. Can you answer that question? Can you pull it together? Even the devil, Google, is pulling together under their supercomputer. It's going to come from some of the evil side of social networks, but that's where the data is. I'd like to hear something like that, because you're not just going to, I'm talking about my cliff, you're not just going to continue to throw money at it. The money won't be there.
Do you want to take it, Dr. Choi? Okay.
Sorry, I think that everyone that I'm associated with is dramatically underpaid, I would say, actually. And our medical students work from anywhere from $0 a year to a very low stipend and things. But I just want to say is that when I thought about being a professor, I thought I would just think and read and talk. But I actually spend about half my time writing grants, and I'll just tell you that the big killer for innovation is, I think, writing grants. And if every one you wrote, you got one, everyone you wrote, you got one, it would be different, but obviously many times you don't get it. And that's kind of one of the reasons I'm here, is because it's just such an important part of our engine of innovation, because we have the time that we've bought to be able to talk and think. So for us, we haven't partnered with Google or these other players, but I just have a really strong bevy of very smart young people who really are passionate about the problem, and that's how we're trying to innovate. So even if it seems like a lot of money, it's actually, I think we're all underpaid. Maybe not me, but all my trainees are underpaid. I wish I could pay them more. But I would say is we probably do it maybe less than 20% of what a pharmaceutical company would pay to do the same thing in terms of all the overhead, salaries, et cetera. So the most efficient way to do it is to give it to academic facilities that can do it, I think. So.
Yes, Dr. Hilsenbeck.
One more comment is that all of us that get grant money are required to deposit data in publicly accessible repositories. This is an innovation that started maybe 15 years ago. It's been growing with the genome, sequencing of the genome. That was the beginning, really, of public deposit of data. When the sequence came off the machines, it went into a public repository almost immediately. That has been an enormous resource. And there have been sequencing and expression sequencing of RNA taking place over the years that are huge resources. Many of the data were deposited maybe a decade ago. They're still being used productively. And there are new requirements and new expectations that even individual experiments get deposited in a publicly accessible and findable way. So there are principles around this called the FAIR principles: findable, accessible, integratable, or interrogatable, and retrievable, right? So they're indexed in a way that anyone can find the data. Now, not all data can be shared absolutely generally, because some of it is somewhat confidential, right? Like, you maybe wouldn't want your sequence to be findable to everybody, but these kinds of deposited data, you can request access, propose a legitimate research need, confirm that you will treat it confidentially, confirm that you will do what you say you're going to do, and so on. So this is a revolution in making data available in the cloud and to researchers who do not have to then repeat these experiments and repeat these sequencing. So Dr. Choi will be depositing his Merkel cell data in a way that other people can find it.
It's already done.
After publication, or some of it's already deposited. The same thing with Dr. Campbell. Data that she's using will be available to other researchers in a way that can be integrated together. This is an incredibly efficient way to do things, because we don't have to repeat these experiments. We don't have to redo these clinical trials in order for the next person to be able to ask these questions. So this is the basis of being able to assemble large data sets that can then do machine learning and artificial intelligence to help us with the next steps in a very cost-effective and efficient manner. So a really important advance in terms of the way the biomedical community works. And it's not just available to investigators in the US. This is a worldwide resource. We use resources from Europe and other parts of the world, and they use ours. So it's a really great way of bringing things together.
Dr. Bast had a comment, and then Dr. Nelson.
Actually, this is a follow-up to the $3 billion in 8 or 12 years, some people estimate, and how we're going to be more efficient with the dollars that we do have. There are many, many steps in developing new targeted therapies or new immunotherapies. We've known about those steps for a long time, but we've got new tools to do that much more efficiently. And we have an experiment going on at MD Anderson. We've got an Institute of Data Science and an Institute of Applied Cancer Science. And the Institute for Applied Cancer Science makes drugs. We're one of the few places where in an academic institution we can actually make new agents to help patients with rare diseases, or to do something that academia thinks about but hasn't gotten to the private sector yet. And with that, we're in the process of putting those two things together. We're on a 4-year experiment to see if we can take every step in drug development and use machine learning and artificial intelligence and other data science applications to make it more efficient, to estimate the most likely thing to succeed to the extent that we can estimate that. We'll have to see how it works, but this is something certainly pharma has used data science to design new drugs in terms of computer modeling of what the target looks like, where the nooks and crannies are, how you develop a drug that will just exactly fit. But nobody's actually systematically looked at every step in the process. That's what we're trying to do.
Thank you. Dr. Nelson?
I just wanted to add to that point that you're making about the incursion of data science into the industry that we have and just what extraordinary opportunities there are. The cost of storing data and moving it around has been asymptotically approaching zero for a number of years. It's one of the reasons there's this incredible revolution. I have to write articles for one of the lay magazines, and the one I did last fall, I was able to find out what's interesting. Since 2022, October of 2022, there have been 521 FDA approvals for artificial intelligence-anchored medical devices. It's already start to dominate medical imaging. It dominates pathology. Several companies scan your slides and make diagnoses. It's moving very quickly, and you see these young people here who are making use of it in their research. I think it's going to move incredibly quickly. That's just my bias.
Thank you, Dr. Nelson. Dr. Pinnaker-Worm?
Hi, thank you. And all the wonderful innovations that my colleagues talked about, but just from a business perspective, and I'm not a business person, I'm in research at MD Anderson, but also engaged in some biotech. Just from a business perspective, why $3 billion? It's the overhead you're paying for failures. This is a very, very tough business. And so now a sustainable business that has to come to bat over and over and over again and to sustain it. You've heard phase 1, phase 2, phase 3 trials. Those on average might be $250, $300 million each to get through. And that's a minimum concern, but just for that itself. And that's after all the super cool innovation and processes to get novel and bold ideas into the patient. But as Bob mentioned previously, only about 1 in 8 actually survive the stringency of phase 1, phase 2, phase 3 trials. And as you get into phase 2, phase 3, that's where you're talking about the $200, $300 million cost. Okay, now it failed. Bam, 300 million. You're in debt, right? Finally, you get that one drug out of those 8 that you get some big sales on, but the quote net price of the sustainability of the process, you've got to now pay for the failures. So from a business perspective, which we've got so many experts here, that's your overhead in order to do it again. Otherwise, you got one shot, you're done, and then you're done, right? So now if you say, okay, the big pharma machine and everything that brings it out, and we want to do it again, and we want to innovate again and again. So it gets back to the efficiency of which we can get better at lowering the overhead of the failures so we can get to the winners. So that's maybe a role for AI and a role for ML and for some innovation now, human on a chip, you may have heard about. Cells and humans that are on little tiny chips. And will the humans on a chip be better than mice and non-human primates in predicting both drugs working and the toxicities around that as better predictors for human disease? Because sometimes the drugs actually work, but the toxicity profiles are too high. So there's 100 reasons for failure, and that's what we're often dealing with. So lots of innovation up front, but yeah, it's an expensive business. Yeah, thank you. Well, I hope so. Let's see if they can all do that, right?
And with that,
thank you.
Somehow we have gotten to 2 hours. Wait, one more short one. One more short one.
Well, I've been thinking about our government and how they spend our money, and right. So I got a better idea. Instead of paying them tax, for them to decide tonight, give a bunch of money, $100,000, and that's $37,000 they won't deploy in the wrong way. So that's my solution.
All right, thank you. And When you see these three wonderful humans, and of course, Dr. Davison, who spoke today, I think you have one message to tell them, and that is, our bags are packed for you. Thank you so much for joining us. I'll see you tonight.